Hello again, this is Tom Gadek. In the third part of the total story of the development of Lifitegrast as a treatment for dry eye. And in this segment, I'd like to tell you about the clinical study of Lifitegrast in dry eye patients. It was studied for clinical signs and symptoms of dry eye in 4 pivotal clinical trials. The first of those clinical trials was phase 1, and the purpose of phase 1 is typically to look at the drug in normal healthy volunteers, all adults, to determine the pharmacokinetics in tear and plasma, show that it's at high levels in tear and low levels in plasma, and then that difference is a safety profile. The pharmacokinetics are also intended in tear to support the dosing of a 5% solution twice a day. 5% dose administered in tear achieves levels that are above 1 micromolar for a large portion of a day. What we also saw in the phase 1 trial was no accumulation of drug in either tear or plasma. So the drug administered is cleared within a 24-hour period or thereabout and doesn't build up over time. So that leads to less concerns about safety. The compound that did get out of the eye showed very rapid clearance in the systemic circulation, never got above about 10 nanomolar in plasma. And that's not enough to affect the function of any T cells. The overall trial showed no unanticipated safety events, no dose-limiting safety was observed. We then, without further ado, I'm going to say goodbye to the phase 1 study trial and move on to phase 2, which is our first effort to demonstrate efficacy in patients. These are all dry eye subjects, and overall, we achieved in this study, in this trial, safety and efficacy. We reached statistical significance in the suppression of corneal staining. That's generating a p-value of less than 0.05. We also saw statistical significance and strong trends in key secondary endpoints that were both signs and symptoms. And the dose response was observed in the efficacy portion. We looked at a broad range of doses crossing 50-fold in concentration and saw stronger response in the higher dose. And overall, it appeared safe in this trial, and there were no unexpected safety events or dose-limiting toxicities. We then moved on to phase 3. There are 3 phase 3 trials, and those are all set up first with the first one called Opus 1. It was designed to confirm the sign that we had seen in the phase 2 trial, and it did. It reached statistical significance for not only corneal staining, but also some key symptoms. And at this time, they were all secondary endpoints. And then again, it appeared to be quite safe. At the end of the Opus 1 trial, Shire Pharmaceuticals came in and purchased SARcode. And at the time, the Opus 2 trial was ongoing, and they ultimately came in, finished out that trial, and then started a final phase 3 efficacy trial that was Opus 3. Those 2 studies, or those 2 trials, confirmed the symptom endpoint, and then combined with the earlier Opus 1 and phase 2 trials, allowed us to file with the FDA for approval for both symptom and sign. The final phase 3 trial was the Sonata trial, which was a long-term safety trial, went out to 1 year, looked at the effect in patients, looked at safety endpoints only. So I'm going to go through each of the individual trials and talk to you about exactly what we were trying to do and why, and what the results were in a lot more detail than I just went into. So the phase 2 trial was the first time we took it into people. We weren't sure what dose to do, what dose to use. So we looked at it against placebo or vehicle, and we looked at 3 different concentrations of drug across a 50-fold range of concentration. In a typical phase 2 trial, the first study in patients, the design of the clinical trial is the key to your success. And we thought we understood Lifitegrast as a potent anti-inflammatory, but we were uncertain what we had seen in vitro preclinically would transfer to in vivo in the clinical trial. In putting together a clinical trial, you need to identify patients. So you'll look at a group of potential patients, and in this panel on the far left, there's 2.5 patients, or 2 severe patients and 1 mild patient, moderate patient, in a population of about 50 people. That's about the relative abundance of patients in the general population, around 5% or so. What we want to do is identify them, group them together into our clinical trial, enroll them, and then treat with drug. And the protocol for the trial shows that they are treated across 3 months. And at the end of 3 months, we then look at that population of what were patients and see what percentage of them have improved and see if any of them have improved back to normal. This is kind of a criteria that the FDA uses, certainly in ophthalmic diseases. They like to see the patients return into the range of normal for any clinical measurement. So the phase 2 trial ongoing, as I said, we were looking to do a dose exploration to establish clinical effect with the compound. We wanted to make sure we looked at a wide enough window to see an effect of the drug. If we study 3 doses that all have no effect, then we're in trouble. We were going to measure both signs and symptoms of dry eye, and we were hoping to be able to get out of this a dose that we could take forward into a phase 3 trial. The signs and symptoms will divide up into secondary and primary signs and symptoms. And in a clinical trial, you have to then specify what your primary endpoints are and your secondary endpoints. The FDA will only consider efficacy in a pre-specified primary endpoint when you go to file for approval of the drug. In the selection of patients, they were enrolled in this trial based on some experience they had with a controlled adverse environment and its effect on both their symptoms and their signs. This shows a controlled adverse environment. It's a room where there's laminar flow of warm air, and that puts stress on your corneal surface. The patients are allowed to then do a visual task, typically watching a movie on a flat panel screen that's slightly above their heads, so they have to look up. And then all this warm, dry air is going over them. They tend to blink less when they're watching a movie, and this stresses the cornea and sets them up for a dry eye event. So it's the equivalent of a treadmill in a cardiac study. It's the equivalent of a treadmill for dry eye. We worked with a company called Aura out of Andover, Massachusetts, that has a proprietary controlled adverse environment room, and we used that for patient selection. And we also use them because the system in which they can run a trial seems to take a lot of the random noise out of the study, and that's very helpful in being able to see a signal. So the part of the design of the phase 2 trial involved the actual formulation of the drug, and in order to be able to see the effect of the drug and the drug alone, we wanted to keep the formulation as vanilla or bland or non-therapeutic as we possibly could, and basically we used water that was isotonic batir, essentially PBS, containing the drug, and we did not want any lubricating components in the formulation. We believe that in the Allergan Restasis trials that the lubricating character of their formulation caused them big problems in the trial where they couldn't see the activity of the drug before about 6 months in patients. And so then, because we're not going to put it in the formulation, we don't want patients using any supplemental lubricating drops during the study. As I mentioned earlier, we needed to pre-specify which sign and which symptom we were interested in, we were declaring for the FDA in each trial. We chose fluorescein staining on a 0 to 4 scale because it looks at defects in the ocular surface. And we believe that that correlates with some of our preclinical in vitro studies on the inhibition of T cells. There are multiple secondary endpoints. So in the first phase 2 trial, we looked at everything we could possibly, cast a wide net. And for symptoms, we focused on the OSDI, or Ocular Surface Disease Index, which is a questionnaire patients get. It's one that's been validated in a number of dry eye trials, and so we stuck with that. And there are other things we did look at as well in terms of patient-reported sensations of burning or scratchiness, eye dryness is another one. And so then the challenge is to pick one symptom and one sign and hang your hat on that. We chose corneal fluorescein staining and the change from baseline to day 84 as the primary sign. And this measures the epithelial barrier function, and the imperfections show up as stained areas on the cornea. It's known in the disease that cytokines from T cells inhibit the epithelial barrier function and can kill individual corneal epithelial cells. And we do know that Lifitegrast can block, from in vitro studies, we know that it can block T cell inflammatory cytokine secretion. So this all made sense. The mechanism of action of Lifitegrast fits in with this clinical measure of the sign. So we went with that. In the phase 2 trial, very interestingly, we saw a dose response where placebo in the white on the left has a certain level of activity in this mean inferior corneal staining. So the inferior cornea is this little pie-shaped piece of your cornea on the lower half of your eye, sort of from 4 o'clock to 8 o'clock, if you would. And what you see here is in the left panel that the placebo has no effect. And then you see a dose titration, 0.1% to 5%. You see an increasing effect, decreasing the level of staining observed. And then if you look at it as the mean change from baseline, which is what's specified in the protocol, you see even better results. And we did reach statistical significance in both the 1% and 5% dose strengths. We then looked at the symptom. And in this case, we're using the OSDI measure, and we've broken it down to a subset of visual-related OSDI subscore within the Ocular Surface Disease Index. We saw data trending with dose for secondary endpoints that were ocular discomfort and eye dryness. Overall, we did decide that, again, this data supported 5% as the phase 3 trial dose. So then, in summary, Lifitegrast in phase 2 trial showed biological effect in patients. That's always heartening. We saw a dose response in signs that was statistically significant for fluorescein inferior corneal staining, so the lower quadrant on your cornea. And we saw strong trends in other signs that did not necessarily reach statistical significance, but included total corneal staining, lissamine, conjunctival staining, and then that Schirmer tear test where they measure the amount of fluid that you can get onto a piece of paper in 5 minutes. Overall, we saw a dose response in symptoms. It was statistically significant for the OSDI subset of visual-related questions on the questionnaire. And then there were strong trends in what's called ocular discomfort. So questions around, how uncomfortable are you before getting your dose and then after several months of dosing. Neither of those endpoints was pre-specified as a primary endpoint. These were for our information only. We didn't see any unexpected safety issues as similar to the phase 1 trial. That's nice. And we decided to take the 5% dose forward into a phase 3 trial. The phase 3 program studied Lifitegrast in 4 separate phase 3 trials. They were Opus 1, Opus 2, Opus 3, and Sonata. Opus 1 through Opus 3 were efficacy studies where we were looking at pre-specified endpoints. All of these phase 3 trials used a common protocol across all of the trials, certainly the Opus 1, 2, and 3 trials, and very little modification of the protocol was done in that. The Lifitegrast Opus 1 phase 3 trial had this common protocol as shown here graphically. There's a period of a wash-in or screening where patients for 2 weeks come in. If it's deemed that they do in fact have the disease, they're sent home with just vehicle or placebo, and they're instructed to administer it to their eye twice a day, one drop in each eye, twice a day. And to do that for 14 days, they come back. We put them back in the chamber, in the controlled adverse environment chamber, and we measure again. To get into the trial, they have to show a difference of 1 point on the scale in worsening of the disease when they go into the chamber. And in this case, they come back 2 weeks later. We want to still see that 1 point difference in symptom and sign on a 0 to 4 scale typically. As they come out of the chamber, we want them to have gotten worse by 1 unit. Then we take them, if they don't repeat, or if they've responded to the administration of just the vehicle, they're cut out of the trial. Patients that meet the enrollment then are given drug and sent home, and they fall into 2 groups, either placebo or 5% Lifitegrast. They're to give themselves drug twice daily as a single drop to each eye for 3 months. And in this study, there were roughly 300 patients in each of these groups. The goal of the trial was to confirm statistical significance that we had seen in the inferior corneal staining in the phase 2 trial, and secondarily to establish statistically significant effect on the primary visual-related OSDI subscale, or on secondary ocular discomfort and/or eye dryness. Those latter 2 are scored on a 0 to 100 visual analog scale. This is typical in a patient-reported study where you'll give them a scale that's arbitrarily, let's say 5 inches long, and they're to draw a line across it where they think their symptom is between 0 and 100. The design overall is identical to the phase 2. And the first results from the Opus 1 trial showed that there was shown in the black bars a decrease in staining of the cornea, inferior cornea, and that indicated an improvement in the cornea barrier function or integrity. We did reach statistical significance, less than 0.05, and similar to phase 2, if combined with the phase 2 results for inferior corneal staining, we now have 2 well-controlled studies that demonstrate statistical significance with a p-value of less than 0.05, and that's sufficient to submit to the FDA. At this point, we could have filed with the FDA and gotten approval for just the treatment of the sign, but we were hoping to get treatment of the symptom as well in approval by the FDA. So again, the Opus 1 trial showed statistical significance in corneal staining, and it confirmed the results from phase 2. In the symptom side of things, we saw that both eye dryness and ocular discomfort improved in over time and with presence of drug relative to the control vehicle or placebo. But we were unable to replicate the results we had seen in the phase 2 trial for the OSDI VR subscore. We were a little dismayed by that. It didn't reach any statistical significance. In the safety portion of the Opus 1 trial, what we did see was, again, no unanticipated or serious ocular safety events, very similar to what we had seen in phase 1. We did look a little more at the ocular safety, and the most common adverse events were a sense of stinging where you put the drug in right at the time that you put the drug on, particularly with the first dose on day 1. It seemed to go away over time with more and more doses. In the trial, 96% of the patients stayed in the trial across the whole 3-month period, which is an indication that the irritation wasn't severe enough to drive people away. Again, tolerated pretty well. The non-ocular side effects or adverse effects included dysgeusia, which is a sense of taste. The drug administered to your eye will drain down from your eye into your nose and then through your tear ducts and then reach the back of your throat where you might actually be able to taste it. The Lifitegrast Opus 2 trial was intended to confirm the statistical significance that we had seen on eye dryness. Again, we think we have the clinical sign of corneal fluorescein staining in the bag. We don't need to show that again. In this case, in the Opus 2 trial, the eye dryness score is now the pre-specified primary endpoint in this study. So it's moved up from a secondary endpoint in Opus 1 to the primary in Opus 2. The results showed here that we were able to see eye dryness separate into statistically significant difference in change in mean from baseline to day 84. So this is the pre-specified endpoint. It confirmed the statistically significant effect on eye dryness seen in Opus 1. So that was very good. And in this case, it's pre-specified. So it's data we can talk to the FDA about. We also saw that the study demonstrated signs and symptoms of dry eye are independent endpoints at this point because we see a divergence with the corneal staining showing an improvement but not reaching statistical significance. So the corneal staining we see as a demonstration of efficacy is judged by mean change from baseline, and we see it as an improvement to day 84, but overall it did not reach statistical significance, so we're not going to be able to use this as part of our package with the FDA as one of two studies that shows statistically significant benefit. However, we think we already have those from the phase 2 and Opus 1 trials. We then looked at safety as well in this Opus 2 trial. Again, as with the previous ones, we saw no unanticipated or serious ocular safety events. The ocular safety concerns included installation site complaints. Again, we had seen that in Opus 1. And these were more common on day 1 than on day 90 at the end of the study. And what we saw also was that report or that discomfort for the individual patients seemed to fade after a few minutes, certainly less than 5 minutes. And it also faded over time as the drug was dosed day after day for 90 days. Overall, the retention rate of patients in the study or in the trial was 96%, meaning that 4% of the patients dropped out. So that's pretty good that everybody hung in there. There were no serious ocular treatment emergent adverse events, so-called TEAEs, nothing related to drug that got to the serious level. And again, on the non-ocular side, dysgeusia was the most common thing, now measured at about 16% of the patients in this trial. Overall, the results are very, very similar to OPUS 1. We went and did OPUS 3, and in this, the goal was to confirm the statistically significant effect on eye dryness from OPUS 2. The design, again, was identical to OPUS 1 and OPUS 2. And the controlled adverse environment was not used for patient enrollment. And in the OPUS 3 trial, what we saw was that eye dryness, as shown here in this table and also these bar graphs, if you would, shows that, in fact, you get a lessening in favor of Xiidra, the trade name for Lifitegrast. You get a lessening of the sense of eye dryness as reported by patients on a visual analog scale from 0 to 100. This confirmed the statistically significant effect that we had seen in OPUS 2, and as reported as a mean change from baseline for the eye dryness score. We also saw that corneal staining did improve as well in OPUS 3, although in OPUS 3, corneal staining was in fact a secondary endpoint. But again, an improvement there. And this confirmed the efficacy that had been observed in OPUS 1 and OPUS 2. Looking closer, and in summary for the 4 trials that we're talking about, the phase 2, OPUS 1, OPUS 2, OPUS 3, shown here, phase 2 upper left and OPUS 3 lower right of this panel, shows an improvement favoring Xiidra, which is the trade name for Lifitegrast, for all 4 of these trials. The mean change from baseline, the dot in these graphs, at day 84 or to day 84, appears left of the horizontal dashed axis. So that's an improvement. It's in the green zone that I've accentuated in the lower left panel. So this is good. In 4 separate trials, we've seen an improvement in the eye dryness score. The first 2, phase 2 and OPUS 1, the eye dryness score was not pre-specified, and in the latter 2 it is. So again, we have now data that we can go to the FDA with that replicates in 2 well-controlled studies the same statistically significant effect in the symptom. Now looking at the sign, which is inferior corneal staining, what we see here is very similar. All 4 trials show a positive benefit for the drug. It reaches statistical significance in 3 of the 4 trials, but the fact that it shows benefit in all 4 trials is impressive. If you were to think about this as a binary event, that it either works or it doesn't work, and you can think about that as a coin flip, if you were to flip a coin 4 times and get heads every time in the green zone here, then the probability of that from your first flip to your last flip is 1 out of 16 times that you do that. You should see 4 positive outcomes. So this is an indication that even the statistically not significant effect seen in OPUS 2 is in fact probably real. We went into a safety trial, and here drug was administered for up to 1 year. The patients were dry eye subjects. The efficacy endpoints, symptom and sign, were not measured in this. We were purely looking for safety endpoints. Interestingly, the installation site irritation continued, also voiced as a reaction. There were some reports as an adverse endpoint that visual acuity was reduced for some patients. And even more interestingly, what you see is that for dry eye, patients getting placebo reported more of a sensation of dry eye than patients receiving drug. Why? That might indicate the drug actually worked. Dysgeusia, again, continued to be the most common treatment emergent adverse event, and there were no overall there were no serious ocular TEAEs. So the drug is looking reasonably safe and efficacious in patients out to a year. This just shows some more data that discontinuations from the trial were slightly higher, 8% versus 5.5% for placebo, and that the common TEAEs that led to discontinuation were similar to what we had seen across the general patient population. Interestingly, if on day 1 or day 0, the first dose of the drug in this trial is given and patients report some discomfort at the installation site, that TEAE is transient. You can see that after 3 minutes, it's down by about 50% in terms of the intensity or the mean comfort score. And then on the last day of the trial, if you look, the overall highest reported or mean comfort score was only 2, so it's down from a level of 3 on day 1. And it follows the same trend that it drops to less than 50% at about 3 minutes. So this discomfort seems to be, the discomfort upon application seems to be transient. Overall, safety summary from the large study incorporating all the trials we've talked about showed that Lifitegrast 5% seems to be safe and well tolerated in dry eye patients for out to a year. There are no unexpected TEAEs in any of these trials and in the larger overarching study. The safety profile, similar to both Opus 1 and Opus 2, continued into Opus 3, and then in the larger Sonata safety trial showed very good safety profile. And the same sort of safety signals came up: installation site irritation and dysgeusia. Withdrawal from therapy was low. People were pretty happy overall, appeared to be pretty happy. Withdrawal from burning is very low at 0.9%. This may contrast to historical data for Restasis, where it's closer to 15 to 20%. And there were no ocular or systemic signs of opportunistic infections. If you inhibit T cells that are vigilant for bacterial or other infections, you might expect that you might see some opportunistic infection. But again, that's just a theoretical concern. And the low systemic exposure we saw in plasma from the systemic sampling showed that, or predicted that there would be a very low risk of opportunistic infection. And we were pleased to see that that carried through. One potential emergent TEAE that was observed progressively across the development and dosing of Lifitegrast is I developed gray hair from worrying about all of this stuff for close to 10 years. So where's Lifitegrast today? I think the science is done. It's been phenomenally successful. We've published as much as we possibly could to try and get this out there. We found that in vitro was predictive of in vivo if we kind of interpreted things correctly. I think our decision that in vitro Lifitegrast could inhibit cytokine secretion and that we expected that to be seen in the clinical sign of corneal staining, measuring the integrity of the corneal surface was in fact true. Mechanism of action helped us to, studies helped us to pick the clinical endpoints. So the FDA approved the drug, thankfully, on July 11th. I was blissfully unaware. The report was they were going to consider approval by July 22nd. And all of a sudden, July 11th, my phone started going off. Texts, calls, all kinds of things from people. Very, very emotional moment. Very gratifying. The FDA granted approval for both symptoms and signs, eye dryness and fluorescein corneal staining inferior. And then Shire has begun marketing the drug as of August 2016. It has the trade name Xiidra with 2 I's. Jennifer Aniston is spokesperson for it. A number of commercials in I Love Patient Education campaign have been noted on television, particularly in the August timeframe. And initial sales reports from the Shire website indicate that sales are strong. It looks like the first month that Xiidra cornered about 16% of the market. That's pretty good for a start. Very early on in the first talk of these 3 talks, this being the third, I said that, you know, the advice that I got very early on from colleagues who I said I was considering starting a company, they all uniformly advised to get a good lawyer. I could not have done this alone, and the greatest burden was carried by Team SARcode members, particularly John Bernier, Chuck O'Neill, Mary Newman, Charlie Semba, and Quentin Oswald and Todd Creech. Not to diminish everyone else's, those people carried a lot of the load. The investors were very, 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 very helpful. They stood behind us. They still stand behind us. And then a number of people gave their time freely along the way to help me work out some of the thoughts that we had. And their advice was invaluable. Their time was invaluable. And then we used a number of contract research organizations who worked with us very, very well, helped us, you know, that we would explain what we were worried about and they would help us get over it. That's it. I think that between these 3 talks, what I've tried to convey is that we did quality science, we published it, and then we used our judgment on data from quality experiments to make decisions on which way to go. In that manner, I believe that scientists can predict the future. And Chuck O'Neill and I, when we sat on the bench at Pete's in 2006 and looked down the road, we saw the approval, at least the way to get to approval. And I think that's the beauty of doing science. You can predict the future. Thank you.